Hello and welcome to this webinar, The Making of Recombinant Anti-Idiotypic Antibodies for High Performance in Bioanalytical Assays. My name's Amanda Turner and I'm the Product Manager for Custom Antibody Products at Bio-Rad. In a moment I'll introduce our speaker. First I'd like to let you know that this webinar will be recorded and an on-demand version will be available shortly after the webinar on the Bio-Rad Antibodies website. Should you encounter any technical problems during the webinar, please click on the question mark at the bottom of the screen. This will bring up the technical guide, which describes solutions for the most common technical problems. And now I'd like to introduce Dr. Christian Frisch. Christian is an R&D manager at Bio-Rad and has led the antibody generation group since 2003. He studied molecular biology in Göttingen, Germany, and did his PhD with Professor Alan Furst at the University of Cambridge in the UK, working on the energetics of protein-protein interactions and protein folding. In 1997 he joined MorphoSys, working on the development of display technologies and the generation of antibody phage display libraries. His group at Bio-Rad has used the HuCAL antibody library and phage display technology to generate more than 25,000 antibodies over the last 13 years for custom projects and for our recombinant antibody portfolio. And now over to you, Christian.
Thanks, Amanda. This webinar is about the making of recombinant anti-idiotypic antibodies for high performance in bioanalytical assays. I will start my presentation with a definition of an anti-idiotypic antibody and then describe the two major assays in which these antibodies are used: immune response or anti-drug-antibody assays and pharmacokinetic assays. I will then introduce you to the three different types of anti-drug antibodies and how these are generated by guided selection using HuCAL phage display technology. I will go on to show several examples for the use of HuCAL antibodies in IR and PK assays. In the last part of my presentation, examples for the quality control and characterization of our biotherapeutics antibodies will be shown. There will be a Q&A session directly after the talk, so please send in your questions already during the presentation. Use the Q&A icon at the bottom of the screen, or send your questions and comments via Twitter using @BioRadApps and #BioRadWebinars. Anti-idiotypic antibodies are antibodies that are directed against the idiotope, which is the unique set of antigenic determinants of the variable portion of an antibody. The idiotope consists of an antigen binding site, which is called the paratope, as well as regions near the paratope which are not involved in antigen binding but are still unique to this antibody. The paratope is the region that binds to the epitope of the antigen. Anti-idiotypic antibodies are specific for one particular antibody, for example the drug antibody. They are inhibitory if they bind the paratope, since then they compete with antigen binding. One of the major assay types where anti-idiotypic antibodies are used are immune response, or for short IR assays, for detection of anti-drug antibodies in serum. Protein drugs are frequently immunogenic and lead to development of anti-drug antibodies, abbreviated ADA. Even humanized and human antibody drugs may elicit a humoral immune response, and these anti-drug antibodies may be neutralizing the drug efficacy or create safety issues. Therefore, for preclinical and clinical development of antibody or protein drugs in general, innovator drugs as well as biosimilars, the sponsor must show the immune response to the drug. And these ADA assays require reference standards, which are ideally human anti-drug antibodies, which cover a broad range of affinities and bind to varying epitopes. In some cases, also different isotypes or antibody subclasses are needed in order to reflect the natural immune response. What is also very important is a consistent supply of the reference standard antibodies over many years. The second major assay type where anti-idiotypic antibodies are used are pharmacokinetic assays for quantification of human antibody drugs in serum. With these assays, the fate of the drug from the moment that it is administered up to the point at which it is completely eliminated from the body can be measured. When a human or humanized antibody drug is administered into a patient, the drug becomes hidden within the high concentration of very similar antibodies in human serum. Typically, the concentration of the antibody drug can be 1,000,000 times lower than the serum antibody concentration, making the drug difficult to detect and quantify. In order to measure drug levels with a pharmacokinetic assay, highly specific and highly sensitive reagents are needed that bind to the drug, but not to the vast excess of similar immunoglobulin molecules. Typical assay formats are shown at the bottom of the slide. The antigen can be used to capture the drug antibody, which is then detected by an anti-Fc antibody. Problems may arise with this assay format since patient serum can contain autoantibodies against the target, and the anti-Fc detection reagent, which is the gray antibody, will react with all human antibodies present in the sample. This may lead to false positive results if an extensive washing protocol is not established. The other two assay formats shown here are preferred since they use highly specific reagents, anti-idiotypic antibodies, for capture or for both capture and detection of the drug antibody. We have classified anti-idiotypic antibodies into 3 types according to their binding modes and properties. Type 1 is the most common class. These are paratope-specific anti-idiotypic antibodies which detect free drug. They neutralize the drug antibody since they inhibit the binding of the drug target. In contrast, type 2 anti-idiotypic antibodies bind to a region outside of the paratope, and they are therefore not inhibitory. These antibodies detect total drug, free, partially bound, and fully bound, and are ideal reagents if there is high concentration of drug target in serum. The third class, type 3, are drug target complex specific antibodies, which only bind the complex and do not bind free drug or drug target, and which therefore detect bound drug exclusively. The anti-TNF-alpha drug antibody Golimumab is an example where we generated all 3 types of anti-idiotypic antibodies. Specificity was probed by ELISA on Golimumab and TNF-alpha Golimumab complex, and different control proteins, including several antibodies with the same isotype, like, for example, omalizumab and adalimumab, which are also IgG1 kappa antibodies. The type 1 anti-idiotypic antibody only binds free Golimumab. The type 2 antibody binds both free and complex Golimumab, but not any of the control proteins. And the type 3 antibody is specific for the TNF-alpha Golimumab complex. It does not bind to TNF-alpha or Golimumab alone. Other assays were performed to further analyze the anti-idiotypic antibodies and to discriminate type 1 and type 2 antibodies. In this example, 2 anti-infliximab antibodies were tested in an inhibition ELISA. The anti-idiotypic antibodies in Fab format were titrated into a constant amount of infliximab. After incubation, free infliximab still capable of binding to TNF-alpha was quantified on immobilized TNF-alpha. Detection was performed with an HRP conjugated anti-human Fc specific antibody. Antibody ABD 17841, shown in black, is a type 1 antibody. It inhibits the binding of infliximab to TNF-alpha. So at high Fab concentration, infliximab binding sites are covered with Fab, and it cannot bind to coated TNF-alpha anymore. ABD 19370, on the other hand, shown in red, which has a similar affinity, does not inhibit binding of TNF-alpha to infliximab and is therefore a type 2 antibody capable of detecting total drug. We also perform epitope binning by measuring real-time kinetics on a 40-bio octet machine. Infliximab was coupled to a sensor which was probed with ABD 17830, another type 1 antibody. After saturation, the sensor was dipped into solution containing the type 2 anti-infliximab antibody ABD 19370, which can still bind to the infliximab ABD 17830 complex, showing that it targets a different epitope on infliximab than the type 1 antibody. The control experiment shows that ABD 19370 also binds infliximab when applied as first antibody. Now, how do we generate these very specific antibodies? We use a recombinant antibody library in the Fab format, which is called HuCAL Platinum. HuCAL stands for Human Combinatorial Antibody Library. The library was made by gene synthesis, so it is not restricted by a tolerance mechanism. There are 7 VH and 6 VL master genes, which cover the structural and sequence diversity of the human immune repertoire. A modular gene design was used for library generation, and high-quality trinucleotide CDR cassettes were used for diversification of all 6 CDRs. It is a very large library containing 45 billion functional human antibodies. For selection of antibodies from the library, we use cis-display, which is a proprietary phage display method where the Fab is linked via a disulfide bond to a phage coat protein, allowing elution of phage with reducing agents. The modular design of the library allows straightforward affinity maturation using pre-made trinucleotide cassettes. I will show an example in a few slides. Antibody generation is done in vitro, so there is full control over the selection process, which takes only 8 weeks. Antigen is incubated with a HuCAL phage library, and 3 consecutive rounds of phage display guided selection are performed. After the 3rd round, the whole pool of Fab genes is cloned into an expression vector, and here there's a choice between monovalent and bivalent FAB formats. Screening is performed in 384-well format using E. coli lysates of expression cultures. Hits are sequenced and unique antibodies are expressed, purified and tested in ELISA or other assays. If needed, the Fabs can be converted into full immunoglobulins. I will now explain which guided selection strategies we use for generation of the different types of anti-drug antibodies. For development of type 1, so inhibitory anti-idiotypic antibodies, selection is carried out on the drug, which can, for example, be immobilized on magnetic beads or an ELISA plate, in the presence of isotype subclass matched antibodies as blockers to avoid enrichment of specificities that bind to the constant regions of the antibody drug. Selection is also performed in the presence of human serum to avoid matrix effects later in the assay. For the selection of type 2 and type 3 antibodies, panning is done on the drug target complex, and blocking is with isotype-matched control antibodies and with target antigen. This drives the selection towards antibodies specific for the complex or to idiotypic epitopes not covered by target antigen. To ensure selection of antibodies with high affinity or to select antibodies with different affinities, an off-rate ranking is performed with the positive clones from the ELISA screening. The K-off screening is done on the Forte Bio octet machine, where it is possible to work with crude Fab containing E. coli lysates. This example shows sensograms of 95 hits from an anti-cetuximab antibody generation. Off-rates measured with crude lysate and purified Fab correspond very well. Sequencing of these 95 clones resulted in 56 different antibodies, so there is a high diversity, and the antibodies cover a broad affinity range from 174 nanomolar down to 500 picomolar. This graph also shows that from K-off ranking we do select the antibodies with the best affinities. The 7 red dots represent the antibodies selected from sequencing the 20 best clones from K-off ranking. If necessary, we can further improve the binding by affinity maturation. In HuCAL, the gene regions coding for the CDRs are flanked by restriction enzyme sites. So for exchange of a CDR, we can use highly diverse pre-made cassettes, which makes maturation very easy. Usually either the LCDR3 or the HCDR2 are exchanged with a cassette, and a new library is generated from the parental Fab, which has variability only in one CDR. This library is subjected to another panning using stringent selection conditions like prolonged washes and decrease of antigen concentration in the panning rounds. An off-rate ranking is performed, and the clones with the desired off-rates are sequenced. The next two slides show an example of the output after standard panning and subsequent maturation of two anti-idiotypic antibodies. From a standard guided selection of type 1 anti-idiotypic antibodies against trastuzumab, several highly specific Fabs could be generated which have affinities in the single-digit nanomolar range. The two antibodies which performed best in PK or ADA assays were affinity matured. These were ABD 16712, which already has a subnanomolar affinity, and ABD 16714. The maturation of the two antibodies resulted in 56 new hits, 26 for ABD 16712, 30 for ABD 16714, and all matured antibodies were shown to be still highly specific for trastuzumab, as shown in the ELISA. Affinity measurements revealed affinities down to 70 picomolar for ABD 16712 derivatives and down to 20 picomolar for ABD 16714 derivatives. The maximum affinity improvement after this only one round of maturation was 120-fold. This example shows that the affinity of antibodies with low nanomolar or even subnanomolar affinity can still be significantly improved using our straightforward, well-established affinity maturation process, which is applicable to all HuCAL antibodies. I would now like to show you examples of PK and ADA assays using different anti-idiotypic antibodies. The first example is a PK assay in the bridging format. Anti-trastuzumab, which is catalog number HCA169, a type 1 Fab with a KD of 70 picomolar, was used as the coating antibody, followed by trastuzumab spiked into 10% human serum as the antigen. Detection was performed with HRP conjugated trastuzumab antibody HCA177, also a type 1 antibody, and this is in the HIgG1 format and has a KD of 20 picomolar. Trastuzumab can be detected down to the few hundred picogram per milliliter range in 10% human serum. An equally sensitive assay was built using the type 2 anti-idiotypic antibodies as capture, and the type 1, so an inhibitory antibody, as detection antibody. In this combination, it is possible to use a high coating concentration, in this case 5 microgram per milliliter, since capture and detection antibody bind different epitopes. Antibodies which are specific for the drug target complex allow to design assays for analysis of levels of bound drug. Here's an example of such a type 3 antibody, which is highly specific for adalimumab complexed with its target TNF-alpha. In this specificity titration ELISA, ABD 18754 only binds to the TNF-alpha adalimumab complex. It does not show any binding to TNF-alpha or adalimumab alone or to TNF-alpha infliximab complex. With this drug target complex specific antibodies, a very sensitive and robust assay was built. TNF-alpha was coated at quite high concentration of 5 microgram per milliliter, and after blocking, adalimumab titrated in 10% human serum was added. Detection was with HRP conjugated type 3 antibody in the human IgG1 format. Also in this assay, detection limit of adalimumab is below 1 nanogram per milliliter. For use of anti-idiotypic antibodies as reference standards for immune response assays, the Fabs are converted into full-length human immunoglobulin, which is a straightforward process. The genes coding for VL and VH are cloned into mammalian expression vectors containing the genes coding for the respective constant regions. We can produce the antibodies as human IgG1, 2, 3, or 4, and also as IgA, IgM, or IgE. Here is an example of an ADA assay using the bridging format. Adalimumab was immobilized on a microtiter plate. After washing and blocking with 5% BSA and PBST, 3 different anti-adalimumab antibodies in human IgG1 format, these are catalog numbers HCA 203, 204, and 205, titrated in 10% human serum were added. Detection was performed using HRP conjugated adalimumab. As expected, the titration curves correlate with the affinity of the antibodies. The 160 picomolar anti-idiotypic antibody ABD 18654 shows the highest sensitivity, and ABD 18664 with 100-fold lower affinity clearly is least sensitive. We see the same thing for other ADA assays. Here is another example with 3 anti-cetuximab antibodies. Again, sensitivity of titration is correlated with affinity of the anti-idiotypic antibody. Before I come to the last part of my presentation, here's a brief reminder to submit your questions. Now I will show you examples for our quality control and characterization of anti-idiotypic antibodies. Every new product is first produced in 3 independent batches. The activity of the batches is compared in a ligand binding assay, ELISA titration for Fab antibodies, and ADA bridging ELISA for IG antibodies. The batch closest to the average curve is chosen as the future reference batch. Every new batch is compared to the reference batch using the same assay protocol. In this graph, the anti-drug-antibody bridging ELISA assay for one of our anti-palivizumab anti-idiotypic antibodies is shown, which is in human IgG1 format. Lots 1 to 4 of this anti-idiotypic antibodies were tested in parallel, and 1 lot was selected as the reference batch. With an error, all lots show the same binding. Before release, new batches of oral antibodies are also routinely tested for specificity, purity, and full-length IG antibodies are tested by size exclusion chromatography for monodispersity. In addition, for each new batch production, the antibody gene is resequenced to ensure product identity. This slide shows the purity of Fab format antibodies assessed by SDS phage with subsequent Coomassie staining. Purity is very good, and heavy chains, or FD chains, and light chains run at the expected molecular weight. 3 different lots of human anti-palivizumab human IgG, which is catalog number HCA262, were analyzed by capillary electrophoresis. Samples were denatured and run under reducing as well as non-reducing conditions. Heavy chains and light chains under reducing denaturing conditions are visible at about 50 kilodalton and 30 kilodalton respectively. Whole IgG molecules, H2L2, are visible at 150 kilodalton under non-reducing denaturing conditions. And these 3 lots show very similar purity. Size exclusion chromatography is used to look for the presence or absence of aggregates. For all 3 lots of the anti-palivizumab antibody HCA262 in IgG1 format, there is basically 100% monomer. Also, the retention time for the 3 lots is almost identical. Antibody shelf life stability can be tested by using an accelerated stability testing protocol or by monitoring activity after several freeze-thaw cycles. In the example shown here, the anti-bevacizumab antibody HCA185, which is a human IgG1, was tested by measuring activity after up to 14 days incubation at 37 degrees. Freshly thawed human anti-bevacizumab human IgG1 was diluted to 1 mg per milliliter, aliquoted and placed at 37 degrees. Aliquots were removed from 37 degrees at every time point and placed at 4 degrees C along with the control. The activity of the samples was measured in a titration ELISA on coated bevacizumab. Even after 14 days at 37 degrees, there is only a slight decrease in activity, indicating a very good long-term stability. 2 independent batches of the anti-adalimumab antibody HCA202 in monovalent Fab format were tested for activity after several freeze-thaw cycles using indirect ELISA. Both lots perform very similarly and do not show any decrease in activity even after 10 freeze-thaw cycles. Similarly, 2 independent batches of the anti-Golimumab antibody HCA204, which is in human IgG1 format, were also tested for activity after freeze-thaw cycles, this time using an ADA-bridging ELISA setup. Also for this antibody, both lots perform very similarly and do not show a significant decrease in activity even after 10 freeze-thaw cycles. We determine the affinity of all anti-biotherapeutic antibodies in their monovalent format using biolayer interferometry. Affinity is the strength of binding between an antigen and an antibody binding site. The equilibrium dissociation constant between the antibody and its antigen is designated K capital D and is the ratio of the experimentally measured off and on rates Kd and Ka respectively. The lower the affinity value, the higher the affinity of the antibody. Examples are shown for an inhibitory anti-idiotypic antibody on the left and a specialized drug target complex specific antibody on the right. We use the monovalent Fab format for affinity determination, as the measured affinity then truly reflects the intrinsic antigen binding affinity due to the monovalent interaction with the antigen. Knowing the affinity of an antibody can give the assay developer an indication of the sensitivity of an assay that might be achieved. I would now like to summarize our custom service for anti-idiotypic antibodies. We offer a fast antibody generation, only 8 weeks for antibodies in FAB format, and the sequence of the antibodies is available. Selection can be driven towards the antigen binding site of the drug antibody by addition of soluble framework-matched antibody, which can block selection of framework-specific antibodies. We can select drug target complex binders or non-paratope-specific anti-idiotypic antibodies. There are no matrix effects of human serum with our antibodies, since we add human serum already during the selection process. Very high affinity can be achieved, leading to sensitive assays. The affinity of the anti-idiotypic antibodies can cover a broad range, which is useful as reference standards for immune response assays, as clinical responses vary and may be of modest affinity. And all of the antibodies can easily be converted into fully human IgGs, IgA, IgM, or IgG, or IgE for use as reference standards in immune response assays. We also offer anti-idiotypic antibodies against 14 drug antibodies in our catalog, which are shown in this table, and we constantly add new specificities. We offer type 1 antibodies for all drugs and type 2 or 3 for some of them. The antibodies cover in most cases a broad affinity range and are all tested in PK and ADA assays with detailed protocols available on our website. To find out more about our anti-biotherapeutic antibody products and services, go to the green resources icon on the webinar screen. Here you'll find links to technical articles, more information about specific anti-idiotypic antibody products, and our custom HuCAL antibody generation services, plus additional webinars on similar topics. Contact us via your local office to talk to an antibody specialist. And now it's time for the question and answer session. Click on the purple Q&A icon and type your question into the box.
First question is from Sarah. Can you provide more explanation of the difference between type 1 and type 2 antibodies?
Yeah, thanks. Very good question. The type 1 antibodies actually bind exactly where the drug target binds. So in case of Golimumab, these type 1 antibodies compete for binding with TNF-alpha. And the type 2 antibodies, they bind to a region outside of the binding region of the drug target. They are still specific for this antibody, but since they do not compete with a drug target, they can bind both the free antibody as well as the complexed antibody. So they can be used to detect total drug. And the type 1 antibodies can only detect the free drug, which in most cases is sufficient if the serum concentration of the drug target is not high.
Great, thanks. I think that also answers Sarah's second question, which was about the type 1 allows for determination of free and the type 2 used for total. Hopefully that answers your question, Sarah. We also have a question from Robert. How well does this method work for the production of antibodies against non-protein molecules, such as common drugs in the molecular weight range of roughly 200 to 500?
Yeah, this HuCAL technology, this recombinant antibody technology, can be used to select antibodies against any target. And we had a number of projects against small molecules already, and this is as successful as traditional technology. You have to consider that a small molecule also for this phage display recombinant technology needs to be coupled to a carrier protein to be used as an antigen.
Great, thanks, Christian. Another question is, will the slide deck be available to the webinar attendees? We will not be making the slide deck available, but there will be an on-demand version of the webinar available, so anyone who missed the webinar or who joined late can listen again. And all the registrants for the webinar will be sent the link to that on-demand version as soon as it's ready. So, Christian, we have another question. You mentioned that you're adding more antibodies. Which ones are coming next?
Yeah, very soon we will launch antibodies actually against denosumab and etanercept. And there will also be new ultra-high affinity antibodies from a maturation for all three types of anti-Golimumab antibodies. And then we are launching an IgE isotype for an anti-adalimumab antibody, which can be then used as a reference standard for ADA assays to measure the IgE ADA response. And there will also be an anti-trastuzumab antibody in IgG4 format to give assay developers more flexibility for the detection step of the PK bridging assay. And then, depending on demand, we may also generate new type 2 and type 3 anti-drug antibodies for the drugs where we do not have these reagents yet.
Okay, and sort of linked to that question, we have another one. I like your type 2 and type 3 antibodies. What is the success rate for generating those? It looks difficult to me.
Yeah, thanks. It's indeed more challenging to generate type 2 or type 3 anti-drug antibodies compared to the type 1 anti-idiotypic antibodies, since there are simply fewer epitopes available. In most cases, we are still successful, but there are then fewer antibodies selected for these type 2 and type 3 antibodies.
Okay, a follow-up question from Robert on the topic of the small molecules. Can you cite any small molecule-specific resources?
Small molecules? Yeah, I'm not 100% sure what that means. I mean, for maybe we should answer that offline.
Yeah, maybe we can see if we have any publications on those kinds of.
Yeah.
Okay, a question from Yang. When we order the antibody for PK analysis, will the information regarding the type of antibody, KD, and recognition site be available?
Yes, that will all be available and is already available on our website. So we publish all the assay data as well as affinities and the format of the antibody for all the ones that were shown in the table.
And what if it is a custom project?
For custom projects, the affinity determination, you need to tell us whether we should do that or not. And then, of course, we will provide you with all the data that you want. We could also, of course, do PK and ADA assays if that is ordered. I mean, many customers do not want to order that, but do it themselves. But we of course happy to also perform these assays and provide the data.
Another question: in your PK assays, you capture using a Fab format and use the IgG for detection. Is it possible to do it the other way around?
Yes, all different combinations are possible. The setup monovalent Fab is captured and labeled IgG as detection antibodies. This worked very well, and we therefore used it for most assays, but also all the other combinations will work.
And I think this could be our last question now. How often do you need to do affinity maturation to achieve the required assay sensitivity?
For the anti-idiotypic antibodies, it was so far not really necessary to perform an affinity maturation. The required sensitivity for PK assays is in the low nanogram per milliliter range, and this has been achieved routinely with antibodies from our standard selection process. But for ADA assays, it may be necessary to have also ultra-high affinity antibodies, and we offer these for some drugs.
Okay, thanks very much, Christian. That's all the questions we have for you today, and thank you everyone for listening to the webinar, and we hope to see you again at our next webinars.